1590, the Zacharias Johnson Okay, these are the magnification of the microscope, different types of the microscope. Mainly, these are the items of the length, breadth, and the diameter objective as multiplied. And actually, it depend on the optical tube length and the focal length of the object, magnifying the power of the eyepiece, total magnification. We discussed the total magnification. Okay, resolution also very important. The minimum distance between the two visible body settles, which is there, can be seen the separate or not the contact with the other eyes. Okay, sorry. So these are the contrast. Mainly, these are the difference intensity between the two objects or between the object and the background. Okay, light microscope. Light microscope, electron microscope. According to our syllabus, light microscope. Next one is the electron microscope, phase contrast microscope. Different types of the microscopes is there. What is the first one is the light microscope. They can divide the using the sunlight or the artificial light. The divide the four types in the microscope: light microscope, bright field microscope, dark field microscope, phase contrast microscope, and the fluorescent microscopes. Okay, there under comes the light microscope, phase contrast microscope, and fluorescence microscope. Electron microscope. These are the using of the TEM and TEM. Sorry, SEM and TEM. My voice is clear. Okay. Next one, electron microscope also TEM and SEM. Transmission electron microscope and the scan electron microscope. what we'll discuss each and everything. And the transmixing electron microscope, especially, they can see in the inner structure also. Inner structure of the organelles, and the next one, different types of the organelles, especially transmission electron microscope. Some is the scanned electron microscope, especially these are the seen the outer structure of the cell organelles. Okay. Bright field microscope is that the produce the dark image against the bright background as a several objective lens. Is that the two types, simple and compound bright field microscope. Simple is the containing the single magnifying lens. Okay, simple microscope is the single as actual. Dissection microscope is the one type of the simple microscope. Only we have seen the only one eyepiece is there. Okay, the contain the only single magnifying lens, dissection microscope. And next one is the compound microscope. Mainly compound microscope, series of the lenses for magnification. Light passes through the specimen into the object lens. Okay, mainly light passes through the specimen into the object lens. Oil immersion lens is the increase the resolution. Mainly have to the 1 or 2 ocular lens. Okay, the resolution is the approximately we are using the 40 nanometer, 40 and 10 100x. Okay, what are the advantages and disadvantages of the compound microscope? Advantage, it is used to the live or the stained cell. For example, in compound microscope, under the compound microscope. Okay, thank you. When compound microscope, especially, we are seeing the some are the, for example, algal mixture. Algal mixture, you see the different types of the algal mixtures in the movable strains used to due to the live and the stained cells. Simple setup with very little preparation of the required in the, these are the advantages. And next one, disadvantages of the compound microscope. The biological specimens are the of not the low contrast need to be the state. For example, you see the bacterial culture, you will then go to the scan and the TEM, cement TEM electron microscopes. The specimens are often low contrast in not to be the state. Staining may be destroyed or the introduce the artifacts. Approximately resolution limit is the 200 nanometer. Okay, 200 nanometer. Next one is the image of the from bright field in the micro, especially the see the under the microscope gram positive bacteria, gram negative bacteria, different types of the bacterial stains. Okay. The dark field microscope produce the bright image objective against the dark background. Optical system is the enhance the contrast and the unstayed bodies, especially. Specimens appearing the gleaming bright against the dark background, especially dark background. Okay, see the requirement of the dark field microscope is the dark field microscope, dark ground condenser, high intensity lamp, and the Fresnel stop. These are the dark optics of the dark field microscope, different types of the optics. What are the uses of the dark ground microscope? Mainly trypanosome, some are especially some of the biological, especially living things in the animals, like some of the animal, trypanosome palladium, leptosporia, Campina endospores, trypanosome palladium. Okay, some of the animals, you see the under the advanced simple setup, it provide the contrast and stained cells living can be viewed. Disadvantage, specimen needs the strongly illuminated, which is the damaged delicate sample. Okay, this is the very important phase contrast microscope. The first one is the phase contrast microscope described the 1930 Dutch physicist, especially Jernicke. 1934, the first described the Jernicke. It is produced the high contrast image for the transparent the specimen. It's a very important phase contrast microscope. Okay, mainly the phase contrast microscope. You will see the examined by normal light microscopy. At the same time, difficult to see the all cell content because the human eye normally detects the contrast microscope. Mainly are the Fred Jernick in the 1934. Fred Jernick is the 1930 described the phase contrast microscope, 1934. What are the advantage? What are the main advantages of the phase contrast microscope? Especially examined the living cells. Okay, we will examine the living cells in the natural state. These are the main advantages. Only living cells can be examined in their the natural state. That are the very important. Okay, especially living cells are the examined in the natural state. The principles of the phase contrast microscope. It is optical illumination technique, which is a small phase shift the light passing through the transparent specimen converted to the contrast to image changes in the image. The light rays in the phase produce the brighter image. Next one, contrast due to the phase. Okay, is the mainly is are the is the principles working the mechanism is the mainly two stage. The specimen examined under the light microscope, two sets of the rays reach in the eye the diffracted. One is the passing through the scattered by the specimen. Another one is the undifferentiated, not passing through the specimen. Two sets of the rays also reach the eye. Mainly, what are two types of the rays? One is the passing through the specimen. Another is the not passing through the specimen. Two types. The light phase can change the produce the differentiate rays the interface with the other rays as resulting. Okay, light rays the fast produce the brighter image. Light rays out the phase form the darker image. Contrast with out of the phase rays. Okay, these are the phase contrast microscope. Mainly, main important is the what are the main uses of the phase contrast microscope is the especially examining the living cells or tissues. Mainly considerable the cell movement, cytoplasmic flow, and any change in the organelle shapes, and they can observe in the phase contrast microscope. Okay. Because mainly these are the request for the phase contrast microscope, annular diaphragm. Next one, phase plate. Okay. Condenser annulus, that the condenser annulus. Next one is the phase plate. What are the functions of the phase plates? Mainly the enhance the phase difference by the retarding the diffracted by front of the one quarter of the wavelength. Reduce the intensity of direct rays and equalizes with the diffracted rays intensity. Okay. Next one is the structure of the diagram of the phase contrast microscope. Phase contrast microscope. Mainly digital camera system also available. Fitting of the digital camera system, observation, transmitter light, biological microscope, especially phase. These are the phase plate. Okay, these are the phase plate. These are the condenser annulus. Okay, mainly they can observation the living cells, the moving, especially the living cells, tissues, the conservable detail, cell movement, and the cytoplasmic flow also they can see in the phase contrast microscope. The image of the phase is the, for example, is the like the some of the bacterial culture. Okay, is the moving of the some of the bacterial cultures. Where, what are the main advantages of the phase contrast microscope? Phase contrast microscope, the visualization of internal cellular component. Okay, for example, in bacteria, in the internal, what type of, especially they can see in the contrast enables the visualization of the internal cellular components. What are the internal components? For example, uh, uh, some of the cellular organelles. Okay, next one, ribosomes. Next one is the internal structure of the nucleus. Okay, and different types of the they can absorb the under the phase contrast microscope. And next one is the diagnosis of the tumor cells also. They can see in the these are one of type of the advantage of the phase contrast microscope. And next one, examination growth dynamics and the behavior with the variety of the living cells in the cell culture. Okay, mainly cell culture, the observe mainly growth and dynamics and behavior of the wide variety of living cells in cell culture. Cell culture. And next one is the studying of the interpreting of the specimens. Okay, these are the main importance of the phase contrast What are the main important mean? Visualization of the internal cellular components, diagnosis of the tumor cells, okay, exam of the growth and dynamics behavior of wide variety of cell, cell culture. Okay, next one is the what are the main disadvantages of the phase contrast microscope? These are the mainly annuli or ring limits the appearances some of the extent which is causes the decrease of the resolution. Next one, not ideal for the thick specimen, only thin specimen. They can absorb the thin specimen. They are the not ideal for the thick specimen. Shade of the and halo effect of may occur. These are the phase contrast microscope. And next one, we'll discuss the fluorescence microscope. Fluorescence microscope. These are the one type of the already we discussed compound microscope. And next one is the phase contrast microscope. Next we will discuss the fluorescent microscope. What are the importance of the fluorescence? Mainly structure and function of the cell organelles. Okay, and other components, especially they can study in fluorescence microscope the structure and function of the cell organelle. Structure and function of the cell organelles. Mainly the fluorescence microscope expose the specimen to ultraviolet, violet, and blue light. Specimen usually the stained with the fluorochromes, and showing the bright image of the object resulting from the fluorescence light emitted the specimen. Okay. What are the principles of the fluorescence microscope? Certain dyes is also called as the fluorochrome. After absorbing the UV rays, rise to the higher energy levels. Rise to the higher energy levels. When the molecules return to the normal state, release the excess energy into the form of the visible light. The fluorescence. What are the visible? The fluorescence. Okay. Mainly, what are the structure and the functions of the cell organelles and the other components. What are the cytochemical staining? They use for the cytochemical staining. The microscope mainly there in the ultraviolet light sources. Okay, mainly these are also called as the fluorescence microscope. Okay, visible light source is the new type of the microscope. Is the ultraviolet light sources. Okay, light ultraviolet source is also called as the fluorescence microscope. What are the principles? We'll discuss the principal working mechanism of the fluorescence microscope works. And the principle of the fluorescence microscope is the light energy of lower wavelength, especially such as the UV rays. Mainly they can use for the such as the UV rays. UV rays. And next one is the such as especially they can use for the UV rays and the absorbed by the specimen emitted the longer wavelengths also. Okay, to select the particular wavelength light is filter called the existing filter. Existing filter. Mainly the UV lamp and the specimen also they can use for the. Next one is the barrier filter also. Another filter also called as the barrier filter. See, this one is the principles of the fluorescence microscope. Principles of fluorescence microscope. These are the certain dyes called the fluorochrome after absorbing the UV rays. Mainly UV rays is very important in fluorescence microscope. What are the main function? Visible lights. These are the UV rays. Ultraviolet rays are the rise to the higher energy level. Higher to the higher energy level. When the dye molecules in the normal state, they release the excess in the energy form of the visible light fluorescence. These are the optical fluorescent microscope, especially optics of the fluorescent microscope. These are the specimen. Okay, these are the objective. These are the detector. Okay, emulsion filter. Next one is the structure of the fluorescence microscope. What are the uses of the fluorescence microscope? Mainly, uh, mainly are the examining the some of the tubercle bacilli, acridin orange or the aramen rhodomine. These are the mainly they can use for the fluorescence. Especially fluorescence means some of the chrome, especially they can apply the some of the fluorescence which are from aramen rhodomine, yellow fluorescence tubercle bacilli. These are the examine the aramen rhodomine, yellow fluorescence tubercle cells. And next one is the my voice is clear?
Yes, sir.
Okay, thank you. Okay, next one. Fluorescence, especially occur in orange. Occurred in orange also. It gives the orange red fluorescence with the RNA and yellow green fluorescence with the DNA. Okay, immunofluorescence. Next one, fluorescence. Main importance is the fluorescence microscope. Chemical nature of the cell walls can be studied. For example, chemical nature of the especially some of the algal members. For example, some of the cyanobacteria. They are especially the cell wall is made of the uh, especially mainly are the peptidoglycans. Next one, some of the different types of the PO5C members also cell wall made of the different mainly. Okay, especially cyanophysi members are the peptidoglycans. Mainly are the study of the chemical nature of the cell walls can be studied. Chemical nature of the cell wall. Okay, outer structure especially. And next one is the identification of the microbes can be carried out through the method of immunofluorescence, especially specific antibodies are the attached to the fluorochromes. Specific antibody. What are the specific antibodies? Especially they can attach to the fluorochromes. The next one, specific DNA sequence can be localized in the chromosome through the method of fluorescence in situ hybridization, in situ fish. Okay, mainly they can study the chemical nature, outer structure of the cell wall. Okay, and next one is the fluorescence. Some of the specific antibody attached to the fluorochromes. At the same time, specific DNA sequence can be localized chromosomes through the fish in situ hybridization. Next one, some are the allopolyploidy. Allopolyploidy. Next one, chromosome belong one gene can be specially identified through the in situ hybridization, GISH. In situ hybridization, fluorescence in situ hybridization, fish. And next one is the genomic in situ hybridization, GISH. GISH also genomic DNA specimens are labeled with the fluorochromes. Mainly, they can use for the fluorochromes. They are the study of the fish and dish. Okay, especially fish mean the fluorescence in situ hybridization. Next one, genomic in situ hybridization. The both are the study of the mainly fluorescence microscopes. Next one, fluorescence microscope. Next one is the electron microscope. Mainly is that the developed with the Nall and Ruska electron microscope. Mainly electron microscope is the co-invented the Nall and Ruska in 1931. Okay, Jerinko in 1934. Mainly is that the phase contrast microscope developed with the phase contrast microscope. Electron microscope is the developed by the Nall and Ruska in 1931. Electron microscope is being the highly energetic electrons the examine object the very fine scale. Mainly are the electrons. These are the mainly what are the mediator in electron microscope are the mainly electron microscope the beam highly energetic electrons into examine the object very fine scale. Mainly they can use for the electrons. That are the very important. Electrons are the transmitted the specimens. Some are the specimens. Okay. Next one is the magnification can up to the 2 million times, while the best light on microscope can be magnified the 2000 times. Okay. Now my only light microscope they can see in the only some are the visible microorganisms. In null and in the electron microscope, especially magnification can up to the 2 million times, while the best light microscope magnify up to the 2000 times. 2000 times. Okay, 2000 times. What are the transmission electron microscope? TEM. These are the two types of the mainly. What are the major principle? Major principle of the electron microscope is the illumination to the higher levels of the magnification and resolution. Illumination source is the mainly tungsten filament, which is the voltage is the approximately 80 kilowatts. Mainly they can use for the tungsten filament. Tungsten filament. Okay, it is become heated about the 3,500 kilowatts and gives the electrons. The electrons are the mainly The filament or the node plate together known as the electron gun. Mainly they can use for the electron gun. Some of the electrons are the lens used in the focusing achieved by the varying current passing through the lens. Okay. Next one is the instead of the image protected either the photographic plate or on a screen. Okay. These are the major principle working of the mechanism of the electron microscopes. Mainly electron guns. The electrons. What are the electrons? Electron beams. Mainly electron beams. Okay. Okay. And the next one is the TEM. What are the TEM? TEM mean transmission electron microscope. These are the very important. Stream of the electrons is formed, accelerated using the positive electrical potential, focused by the metallic operator with the electromagnets. Interaction occur into the related to the sample, which is the detector transmitted to the image. Okay, see, for example, these are the 2D images, projector lens from the image of the fluorescent viewing the screen out. Actually, the magnification is the approximate 2000. They can see in the TEM internal structure of the DNA, okay, internal structure of the ribosome, internal structure of the some of the cell organelles, internal structure of the cytoplasm. They can study the all type of mainly the inner structure. They can study into the inner structure of the cell organelles. The mainly electron TEM. Next one is the TEMS. Next one is the scanning electron microscope. The scan is the gold-plated specimen give the 3D picture. Already TEM is given to the 2D picture. The scanning electron microscope SEM also they can give the 3D view of the surface of the object, which is the black and white. Use the study of the surface features, cells, and the viruses, especially SEM and TEM are the they can study to the study of the surface features of the cell and the viruses at the same time. The outer structure of the bacteria, mostly they can use for some of the outer structure of the microorganism they can studied by the cement TEM. Okay, cement TEM, outer structure especially. Okay, scanning electron microscope as resolution is the 1000 times. The TEM also resolution is the 2000 times. TEM mainly, what are the resolution power of the TEM? 2000 times resolution power. Okay, these are the topics of the SEM. Okay, SEM images. These are the SEM images. For example, see the Vibrio cholera, Rhodosudomonas, Trypanosoma palladium. These are the see. This one is the flagella structure. They can see in the flagella outer structure. Mainly SEM is the scanned electron microscope. Especially they can study the outer structure. That are very important. Inner structure studied by the TEM, outer structure studied by the SEM electron micrograph, scanned electron micrograph. Okay. Okay. What are the difference between the TEM and beam? SEM is that the very important your annual examination point of view. Okay. Mainly electron beam. Broad statistic beams are that they can see in the TEM. Okay, electron beam. Mainly in the beam of the focused, especially sample scanning line, the electron beam, especially in TEM, broad statistics beam. They can see the broad statistics beam in SEM. Beam is the focus to the fine point of the sample scanned by the line. Okay. The next one is the voltage needed. How much of the voltage is needed into the in TEM? TEM is voltage range from to 60 to 3 lakhs volts. 60 to 3 lakhs volt. In SEM, accurate the voltage much lower, not necessary for the penetrate in the specimen. Actually, the TEM is the very costly electron microscope. The TEM especially are the study, especially they can see in study of the internal structure. Study of the internal structure TEM. The SEM is the only outer structure. They can study the outer structure. Okay. The high volt, they need for the high voltage. Please, who is the Raista? not correct way. Okay. Rajastha, please remove the okay voltage. Next one, the interaction of the beam electrons. These are the very important. Specimens must be the very thin, especially in TEM and SEM. The specimen is the very thin. At the same time, some of the in SEM the specimens are the coated by the gold coated. Okay, the very important. The specimens are the gold coated specimens. Okay, specimens are the gold coated in they can see in the in SEM. Okay, specimens are the very thin. Most of the seen under the microscope. The specimens are the most of these are the very the thin section. They can cut the very thin section. Imaging electron must passes through the be transmitted by the specimen. Transmitted by the specimen. And next one is the in SEM information. It is the collected by the surface of the specimen. The mainly imaging they can transport the inner level. Electrons are the pass through the by transmitted by the specimen inner level. Okay. And next one information is the needed as the collected by the surface of the specimen. Surface of the specimen, especially they can see in the in scanned electron micrograph. The inner level they can see in the imaging of the TEM. Okay. Next one, image reducing transmitted electrons correctly focused by the objective lens magnified to create of the real image. Beam is scanned along with the source of the sample build of the image. Especially especially in TEM and SEM also, the specimen coated by the some of the gold coat. That's very important. Both the specimens are the coated by the gold coated. They can see in the inner or outer structure. Mainly the mainly the outer structure is the they can see in by the scanned electron microscope. The inner structure they can study in the mainly inner structure, especially they study in the TEM electron microscope. There the difference between the TEM and the SEM. And another importance. What are the major difference between the light microscope and the TEM? Mainly, light microscope is the visible light is the source of the illumination. Okay, visible light is the mainly source of the illumination. And next one, TEM is the electron beams are used. These are the very important. Mainly, electron beams are used in TEM. Okay, electron beams are used in TEM. And next one is the light microscope is the glasses lens are the used in TEM. Electron magnetic lenses are used. Mainly electron beams and the electron magnets also they can use for in the transmission electron microscopes. My voice is clear?
Yes, sir.
Okay. Sir.
Yes. Okay. Okay. Light microscope, difference between the light microscope and TEM. Main TEM is the mainly they can use for the electromagnetic lens are used. Electron beams, electron magnet. Okay. The thin section is coated by the gold. Next one, tungsten. Different types of the some of the heavy metals are the coated by the TEM and SEM. Okay, next one is the focal length of the lens is the determines the magnification and resolution. Focal length lens is the determines the magnification and resolution. Strength of the current is the determines the these properties in the TEM. Okay. Next one is the differential light absorption by the parts of the specimen produce the differential images. Mainly differential scattering and the transmission of the electron produce the differential images in TEM. The specimens are mounted on glass slide. Specimens are the mounted on metal grids. What are the metal grids? Gold. Okay. Mainly they can use for the specimens are mounted by the metal grids. What are the metal grids? Mainly they are the gold in temarsum. Okay. In the gold. Okay. Next one is the The chemical dyes are used in the for staining. For example, see the under the microscope, compound microscope, saffronin, next one glycerin, next one is the methylene blue. Different types of the dyes are used in the under the compound microscope or the light microscope. Okay, in heavy, mainly in TEM, in TEM and SEM is the TEM, especially heavy metal coating used instead of the staining. What are the instead of the mainly what are the heavy metals? Gold is the heavy metal. Okay, mainly heavy metal coating is used the instead of the staining. Mainly staining instead of the staining. What type of the metals are used in the in coating are the mechanism? Next one is the there is no need of the vacuum environment in the microscope, light microscope. The vacuum is the very important in necessary to the compound, TEM and SEM. The TEM and SEM electron microscope. Okay, electron microscope. Next one, dehydration of the specimens is not necessary in compound microscope or light microscope. The dehydration is the necessary, the dehydration the specimen is very necessary, is compulsory of the in TEM and scanned electron microscope. Okay, dehydration is the dehydration totally dehydrate the specimen. You see the under the microscope, the total dehydrated specimen. You see the electron microscope. Okay, next one is the maximum resolution is the possible is the in microns approximately 10 to the power of minus micrometers, and the maximum resolution possible into the range of the nanometers is the 10 to the power of 9 in TEM electron microscope. What are the major uses of the some of the microscope, mainly other microscope, these are the confocal laser scanning microscope. There are also the specimens we can see in the only our syllabus is the TEM, STEM. Scanning probe, microscope, scanning system. Okay, this is the microscope. Already, what type of the compound microscope, bright field microscope, fluorescence microscope. The next one is the electron microscope, phase contrast microscope. In phase contrast microscope, especially they can study the living things. Okay, fluorescence microscope, especially they can use some of the fluorescence chrome. Okay, these are the function of the cell organelles and the component cytochemical studies. Okay, electron microscope also mainly electronic beams. Next one. Okay, next topic is the cell organelles, ultra structure of the cell and its organelle. Okay, next topic organelle.
Sir, we cannot see the screen.
Okay.
Hello.
This is the cell. Yes.
Sir, we cannot see the screen, neither your voice is clear.
This is the cell organelles. Previous topic is the microscopes. Are you not seen? Microscopes. Okay, this is the cell and structure of the function of the cell organelles. What is the meaning of the cell? Mainly structure of the cell is the structure. Mainly the basic unit is the life is cell. Okay. Uh, Types of the cells is the two types of the cells, the prokaryote cells and the next one eukaryote cells. Lack of the nucleus is the membrane bound structure is the some of the prokaryote cells, cyanobacteria, bacteria. Okay, mainly only lack of the nucleus. Okay, eukaryote cells is the okay prokaryote the lack of the nucleus, especially bacteria. Eukaryotes is the some of the plants and animals, some of the fungal members also. Eukaryotes have the nucleus and the membrane bounded organelles. Okay, these are the prokaryotes. Prokaryotes are the bacterial cells. Bacterial cells. Some of the few internal structures also in the bacteria, especially bacteria 70S ribosome. Next one, NAG DNA. Next one, plasmids. Okay, the plasmid, some flagella, outer structure, the structure of the cell. Mainly, okay, this is the structure of the cell. Okay, internal structure of the plant cell is the mitochondria, ribosome, plasma membrane, nucleus, ER, endoplasmic return, rough ER, smooth ER, vascicles in the cytoskeleton, peroxisome, lysosome, Golgi operators. Mainly, the first one, the term cell was coined by the Robert in 1665. Okay, and the next one is the popular cell theory proposed by Slide and Schwann. Okay, next one is the will show mainly that the extend the cell theory, especially 1839. Slider and Schwann is the developed by the cell theory, which also they can develop the in 1858 they extend the cell theory. What are the main cell size, shape, and number? Approximately the biggest, especially the diameter of the ostrich, the XL is the 5 to 6 inches. According to our the biggest, the especially the biggest, the total planting kingdom, the total planting. What are the biggest ovules in the cycas? The biggest ovules are seen in cycas. At the same time, cell size, shape, and number. This ostrich XL is the approximately 5 to 6 inches the diameter. Some are the single celled algae. Establiria is the size of the few centimeters. And algae establish approximately some centimeters of the diameter. Okay, some are the shapes of the bacteria, different as the rod shape, cockide. Next one, bacillus, cockide, vibrio. The next one is the streptococcus. Different types of the bacteria also cell size. The biggest bacteria, what are the biggest bacteria? Navibiansis bigiata, especially big Navibian, bigiata, Navibians is the biggest bacteria. Okay. Next one, mainly internal structure, especially the internal structure, they can see in the nuclei envelopes, membrane bounded organelles. Some are the genetic, what are the genetic materials? DNA, RNA, nucleic acid. Next one is the what are the major difference between the plant and the animal cells? Next one is the mainly plant cells have the cell membrane is the some of the made of the mainly plant membrane is the uh, cell membrane especially is made of the uh, mainly the plant cellulose. The outer structure is the made of the cellulose. Okay. The next one is the difference between the prokaryotes and eukaryotes, the types of the ribosomes also. The next one, what are the major difference between the DNA, RNA? DNA, RNA also the different deoxyribose and the ribose sugars. Some are the purines, adenines, different types of the purines and also structure also they can different in the nucleic acid, DNA, RNA. Okay. Mainly cell wall. The cell wall of the bacteria, especially the made of the peptidoglycan. For example, algae cell wall is made of the cellulose. Fungal cell wall is made of the chitin. Bryophyta and pteridophyta, and next one is some of the made of the cellulose. Especially some of the algal members also the cell wall are made of the silicon dioxide. For example, Cara. For example, Cara. Next one, in equisetum outer, especially the outer stem is made of the some of the silicon dioxide, silicon dioxide. Some of the especially gymnosperms and angiosperms, the cell wall is made of the some of the plants, especially made of the cellulose. Okay, especially gram negative bacteria and gram positive bacteria. Gram positive bacteria, especially they contain the some of the teichoic acid, peptidoglycan along with the teichoic acid. In gram negative bacteria, teichoic acid is the option. So teichoic acid are the option. Mainly polysaccharides, especially cellulose. Okay, cellulose. Okay, these are the cell wall. What are the cell wall in plants? Is the three types of the middle lamella, primary lamella, and secondary lamella. They can see in the some of the tertiary wall also. They can see in the cell wall in plants. Mainly is the middle lamella. What are the importance of the middle lamella? Mainly, the middle lamella makes first appearance of the cytokinesis of the cell division. The appearance in the cytokinesis of the cell division. Next one, primary wall is the mainly either side is the middle lamella. It constitutes mainly the made of the cellulose, some of the microfibrils, xyloglucan chains, and next one is the polysaccharides. The peptide polysaccharide also is made of the primary wall. The secondary wall, especially inner secondary. Okay, secondary wall is also some, for example, lignins, different types of the lignins. Some are the lignins, waxes also. These are made of the secondary cell wall. Secondary and tertiary wall also. These are the different types of the some are the secondary wall lining plasma membranes. Some are the degenerated lignin plasma membranes also considered separated by the tertiary wall. What are the ultrastructure and the chemical components of the cell? Is the mainly the cell wall is is made of the most of the cellulose. The cellulose microfibers along with the hemicellulose and the pectin, and the ground substances matrix, which is the cellulose and the microbials embedded. Next one is the like some chemically cell wall is found to consist of the components of the cellulose, hemicellulose, pectin, lignins, some of the proteins. Okay, different types of the outer structure is made of the proteins and the cellulose different. And the cellulose and the hemicellulose, the both are the substances present of the outer structure of the some main is the plant cell wall, the cellulose and the hemicellulose, the outer structure mainly. The main chemical component of the cell wall, cellulose is one of the substance present in the especially air. At the same time, cellulose molecules consist of the at least 500 glucose residues, 1,4 beta glucan chain, mainly 1,4 glucan. Okay, cellulose. Next one, hemicellulose also linking glucans of the called hemicellulose, mainly cellulose crossing cellulose, especially long cross-linked glucans that make the hydrogen bonds with the surface of the microfibrils. These are the most of the cross-linking glucans is called as the hemicellulose, and two such glucans in the higher plants is the xyloglucans. The two higher plants is the two such glucans, the higher glucans is the xyloglucans. Okay, like the pectins also. These are the pectins also. They can see in the higher plants, especially outer structure, they can see the pectins. Pectins also heterogeneous groups. These are the branched polysaccharides. Okay, these are the charged the galactonic acid residues. Lignin also phenolic derivative, the aromatic polymers abundant to the secondary walls. These are the made of the secondary walls. Next one is the lignin. Next one, proteins. Proteins are the primary cell wall. These are the is made of the protein, especially they can see in the outer structure, primary cell wall. Cell wall deposed in the cell shape, especially cell wall is the made of the already cellulose. Most of the plant cell walls is made of the cellulose. What are the functions? What are the functions of the cell wall? What are main functions is the provides the mechanical strength. Provide the mechanical strength, resistance of the tensile stress, stress relation and the cell expansion. Okay, stress and the relation expansion, cell expansion. These are the cells, structure of the cells. Okay, the next one is the nucleus. These are the prominent large organelle. Some of mainly these are the most of the their nucleus they are containing the DNA, contain the genes, control the activity of the other organelles. What are the main function of the nucleus? Controlling the other organelles. Structure of the nucleus. These are the oval shaped structure. And next, most of the cells are the single nucleus. Some are the mainly RBS, and some of the such as the mature RBS have the number of nucleus is also present. Next one, the nuclear envelope. Membrane of the nuclear is also contained with the rough ER. Okay, rough ER. Nuclear poles also present in the nucleus. Each nuclear pore consists of the circular arrangement of the protein surrounding the large central opening. These are the nucleus. Okay, next one is the ribosome. Ribosomes mainly are the synthesis of the protein. The main important, what are the importance of the ribosome? Synthesis of the protein. The also name of these are the tiny RNA, the higher content of the rRNA. And next one is the attached to the rough ER, especially rough endoplasmic reticulum. Next one is the consists two subunits. One is the larger subunit and the smaller subunit, Svedberg units. Mainly, most of the prokaryotes containing the 70S ribosome. 70S ribosome. 70S ribosomes mean 60 plus 40. Sorry, 50 plus 30 is the 70S ribosome. Next one, 80S ribosome, 60 plus 40. The 80S ribosomes, especially they are containing some of the eukaryotes. Most of the prokaryotes, they are containing the 70S ribosome. These are the ribosomes. What are the importance of the ribosome? Ribosomes, especially the name is given by the Palade. Mainly importance of the protein synthesis. Mainly, they are the main importance of the protein synthesis. They are the containing the high amount of the rRNA. High amount of the rRNA. Next one, endoplasmic reticulum. The network of membranes from the flattened sacs. There can be two types of the endoplasmic reticulum. Rough ER, smooth ER. What is the importance of the rough ER? Rough ER, especially smooth. These are the main protein synthesis. Rough ER is main importance is the protein synthesis. Okay, nuclear membrane, rough ER, smooth, and mainly what are the importance of the smooth ER? Smooth ER, they are functioning of the lipid synthesis, protein synthesis, and the lipid synthesis. Rough ER is the mainly synthesis of the proteins. Rough ER involved in the post-translation modifications such as the glycogen. Next one is the smooth ER synthesis of the phospholipids, cholesterol. Next one is the steroid hormone, some of the steroid hormone. Smooth ER is the site of the glycogen metabolism. These are the very important. Smooth endoplasmic reticulum is the site of the glycogen metabolism. Mainly, smooth endoplasmic reticulum is site of the glycogen metabolism. Glycogen metabolism. Okay. What are the smooth ER? Especially smooth ER is the site of the glycogen metabolisms. Next one, storage release the calcium ions in the cells. Liver, kidney, mainly in liver, kidney, smooth ER, the detox with that drugs and the harmful substances. Remove of the phosphate group from the glucose 6 phosphate and the release of the glucose in blood. Ecology complex. It consists of the 3 to 20 cystine, a small pattern membrane sacs is the Golgi complex. Golgi complex is the more extensive the cell that they can secrete the some of the proteins, modify the sorts and the package transport proteins received from the mainly RAFIA, RER, Golgi bodies. Okay, that the processing package the proteins from the Golgi complex. Next one is the mitochondria. Mitochondria are the rod-shaped plot and also called as the powerhouse of the cell. Mainly 100 of the what are main important mitochondria are related to the respiration. Active cells of the some of the muscles, liver, kidneys, which are ATP synthesis. Large number of the mitochondria. The outer structure, inner structure, especially the mitochondria, the respiration, glycolysis, and the Krebs. Especially glycolysis occurs in the cytoplasm. Krebs also in the mat cistern. Okay. Um, Mitochondria. Next one, lysosome. Lysosome are the membrane vesicles. Actually lysosomes, and next one is the ribosomes. Is the both are the unicellular organisms. The outer structure. The mitochondria, chloroplast. Next one, endoplasmic reticulum. These are the double membrane models. Okay. Lysosome. What are the mainly what are the importance of the lysosome? Digestive enzyme, hydrolytic enzyme. They are containing the powerful digestive and the hydrolytic enzymes. Lysosomal enzymes also help the recycle from the outer cell structure. It is the helps the fertilization. Mainly some disorders caused by the failed absentee of the lysosomal enzymes. Okay, lysosomal enzymes. Peroxisome is mainly what are the importance of the peroxisome is also single membrane model. Is the mainly the detoxification process. They can use for the detoxification process. Is that the lyso smaller size? They contain several oxidase enzymes can oxidize the various organic substances. What are the main functions of the peroxisome? Mainly degradation of the amino acid, oxidation long chain fatty acid, oxide toxic substance such as the alcohols. Mainly protect the parts of the cell parts toxic effect. Mainly detoxification. Okay, this is a very important detoxification. What are the main importance of the peroxisome detoxification? And the cytoskeleton is also network of the protein filament that extend the cytoplasm, microtubules, intermediate filaments, the microfilaments, cytoskeleton. Next one is the plasma membrane. Plasma membrane also is very important. Uh, Mainly the chemical composition of the plasma membrane. Membrane lipids, especially the outer structure, especially the membrane is the boundary layer of the cell, separated by the inner contents and the outer cell wall flow. Plants and the extracellular matrix, the animal cells also. Plasma membrane. Plasma membrane is the chemical composition of the plasma membrane. Is the mainly proteins, lipids. And next one is the oligosaccharides. 20% of the water they are contain the mainly proportion of the lipids, proteins are the found the very greatly into the various system. What are the membrane lipids? Phospholipids is the very important. Two different kinds of the phospholipids, the membrane lipids. One is the pH phospholipids, the acidic phospholipids. Okay, pH phospholipids and acidic phospholipids. Next one the phospholipids, glycolipids, cholesterol with the relative proportion also mainly is made of the membrane lipids, phospholipids. Membrane proteins, plasma membranes are the contain the membrane proteins also. Approximately 25 to 75% of the 70% of the membranes are there containing. Mainly are the what are the functions actual the membrane production, mainly membrane production. Okay, proteins mainly lipid molecules are the smaller than the protein molecules. Lipid molecules are the smaller than the protein molecules. Models of the membrane structure, especially Daniel Davidson, Singer and Nicholson fluid mosaic model. Okay, bimolar model. Different types of the models also available. Explain the plasma membrane models. Mainly models of the membrane structure. Mainly first one is the three-layered membrane is called is the unit membrane. These are the developed by the Robertson. Three-layered membranes. Okay, three-layer membranes is called also unit membrane. The developed by the Robertson in 1959. And next one is the supported by the concept Daniel Davidson. Okay, Robertson. Next one is the organization of the lipid bilayer. Mainly lipids and the proteins are the of the plasma membrane. Next one, fluid mosaic mode is the mainly the development 1917 to in Singer and Nicholson. Mainly bimolecular models developed by the Daniel Davidson, and trimolar model developed by the Robertson. Okay, micellar model they can develop with the Hiller and the affinity. Okay, what is the fluid mosaic model? Especially the developed by the Singer and Nicholson in 1972. Singer and Nicholson. Uh, Mainly functions of the plasma membranes also. Membrane transport the proteins. Next one, active membrane transport. Okay, next one are the plasma membrane structures. Lysosomes, also endoplasmic reticulum, peroxisome. Next one is the glyoxysomes. What are the importance of the glyoxysomes? Also, plant species, especially they can store the lipid materials, especially especially in the seeds are the they can containing the some of the lipid materials. These are the mainly glyoxysomes, mainly storage lipid materials in their seeds. Okay. Next one is the main glyoxysomes, especially some of the plant species store the lipid materials in the seeds. Glyoxysomes, for example, main functions of the glyoxysome is with the beta oxidation, glyoxolate cycle. Glyoxolate cycle is the C2 cycle. Mainly beta oxidation are the involve the conservation of the fatty acid to the fatty acyl coenzyme. Okay, oxidation to 3-keto acyl to the finally producing the acetyl coenzyme A. These are the acetyl coenzyme mainly these are the for example in Krebs cycle glucose they can convert the pyruvic acid. Pyruvic acid they can convert the acetyl coenzyme. Acetyl coenzyme they can enter in Krebs cycle. Beta oxidation in between the in between the glycolysis and the Krebs cycle into the beta oxidation process is also available. Mainly they can form the acetyl coenzyme A. stale coenzyme A. And next one is the glyoxolate cycle. The glyoxolate cycle pathway involves the some of the reaction of the TCA cycle, that is the mainly citrate isocitrate form. Okay, the cycle involves the enzymatic reactions. Mainly, they cannot report it in animal cells. Only they are reported to the some of the especially plant cells. Mainly isocitrate, they can convert the glyoxolate succinate enzymes, and they can involve the enzyme isocitrate enzyme. And okay, these are the glyoxosomes. Mainly, what are the importance of the glyoxosomes? Mainly, glyoxosomes are the mainly important is the main storage of the lipid particles, especially for example, resinous communicus. Okay, resinous communicus is the mainly the storage of the oil forms, especially lipids. Okay, vacuoles. What are the importance of the vacuoles? Uh, vacuoles, especially, these are the spherical ovoid structure enclosing space of the covered by the tonoplast. Okay, mainly vacuoles are taken in the some of the animal cells. Vacuoles mainly, uh, sorry, some plants, especially they are in the vacuoles are the plant cells. The centrioles and the centrioles in the animal cells. Vacuoles are the spherical ovoid structure. Vacuoles are the constitution most of the plant cells is the 30% of the 30% of the cell components vacuoles. Vacuoles are this especially inner structure, especially they contain some of the enzymes, organic substances, sugars. Okay, there are many secondary metabolites also they contain the vacuoles. What are the functions of the vacuole? Vacuoles are the multifunctional components. These are the continuous movement of the solutes both direction the vacuole. There is the possibility of the accumulation, next one uptake, and next one the transport of the some of the particles, especially. Main with the multifunctional components. Okay, continuous movement of the solutes, the both the directions of the vacuoles. Next one, plants are use the vacuoles the produce the large cells of the cheaply, especially they can produce the large cells. Mainly, some are the nitrogen components, rich blue hydrocarbon, the vacuoles, especially the V type, H type, ATP, the vacuolar type, or some are the solids like calcium, sodium, calcium, potassium, chlorine, next one phosphate, nitrate ions also they contain the vacuole. Next one is the storage function. These are the very important. Storage function is the vacuole is the very important. Storage function and role in digestion. Digestion also is the very important. The next one is the control pH and the ionic homostatistics. The next one is the defense against the microbial pathogens and the herbivorous. Storage of the pigments. These are the mainly anthocyanins contained in the vacuoles are the common feature of the most of the plants. Anthocyanin. For example, tomato plants. Tomato is the outer layer is especially is made of the anthocyanins. Okay. Anthocyanin, especially anthocyanin contain the vacuoles are the common feature of the most of the plants. For example, the tomato plant outer structure, especially they are made of the anthocyanin. Okay. At the same time, the some of the leaf pigments screening out of the UV visible light to protect the photosynthetic apparatus against the photooxidation. These are the main importance of the vacuoles. ATP, chemical composition of the mitochondria, or the role of the glucogenesis, biogenesis, role of the amino acid in lipid metabolism. Chloroplast also is the main. The term is kind by the shrimper. The plastic, the bimembrane models, these are the shrimper. Okay, mainly chloroplast related to the photosynthesis. Fluorescent color. All types of the plastics also developed. Some of the protoplastids. Mainly are the precursor structure of the protoplastids. Protoplastids. Okay, proplastids are the precursor of the plastids. Mainly, the protoplastids are the appears informed in the finally granular. It is also called as the stroma, lamella with the vesicles. Vesicles. Bio is the plastids, plasma desmata. What are the mainly plasma desmata are the interlinking connecting with the one organism to another, especially plasma. Uh, Mainly are the plasma animals. Some of the animal cells are the containing the plasma desmata. Okay, these are the mainly is are the rich of the cellulose and the eliminate of the some of the mainly junctions in between the cells. They can connect with the one cell to another cells. Mainly connect with the adjusting of the mainly these are the made of some of the main rich of the cellulose plasma desmata. Okay, this is the cell organelles. Okay, cell organelles. These are the two topics is the structure of the microscope, reduction of the microscope, the types of the microscope, types of the cell organelles. Okay, this is the uh, cell
అవ్వదు ఇంకా చాలు తర్వాత సాల్ట్ వేసుకోను పదండి అందులో ఉన్న నీరు పోవాలని
என்னது எழுதிக்கிறேன் போய்
Okay, next one, Samadha. Some are the membrane proteins also. These are the membrane proteins. Okay, okay, membrane proteins. Membrane proteins are the membranes are the in separation of the cell and separate inside of the from outside. Okay. From our eukaryotes membranes also different the intracellular organs such as the nucleus, mitochondria, some are the lysosome. Mainly, some of the proteins of the all cells of the recognize chemical signals and the adjusts the metabolism and pattern of the gene expression of the response. And some of the eukaryotes also the plasma membrane with the multiple that perform the variety of the functioning, including the membrane transport and the signaling of the connecting of the cell membranes. Okay, that the eukaryotic cells have the some of the internal membranes bounded in the organelles, especially eukaryotes, the unique component of some of the protein that the enabled to carry out of the main ATP generation and the ADP DNA synthesis. That the internal membrane bound organelles and the unique component of the protein. Internal membrane bound and next one is the component the ATP generation and the DNA synthesis. And the next one the lipid bimolar. Already lipid bimolar is the composition structure of the organization. The most common phospholipids are the phosphoglycerides. All phospholipids are the amphiphloic, especially phospholipids are the amphiphloic. Both the side the phosphorus there contain the amphiphloic condition. The next one is the bimembranes also contain the small amount of the other amphiphloic lipids, such as the glycolipids and cholesterol. Amphiphloic, what are the amphiphloic nature of the phospholipids? The critical structure of the bimembranes. Critical structure of the bimembranes. The phospholipids are the aggregate of the three forms. One is the spherical micelles in the liposomes and the phospholipid layer, bilayer. Main structure of the depends of the several functions, the including of the fatty acyl chain, hydrophobic entail their degree of the saturation and temperature also the depending on the phospholipids aggregation, the three forms. Main, what are the three forms? Phospholipid bilayer. These are the membrane lipids, especially some of the membrane lipids, phospholipid bilayer, liposome, the spherical micellus. These are the structure of the including length of the fatty acid chain, the hydrophobic tail, the degree of the saturation temperature, the effect of the cause of the acetyl chains aggregate, exclude the water molecules from the core. The main important is the spherical micelles. Are the rarely formed from the natural phospholipids. Okay, nearly formed the natural phospholipids. These are the formed one of the two fatty acetyl chains, and to make the tail of the phospholipid, remove the hydrolysis. Okay. Next one, each layer is the lamellar structure is also called as the leaflet. Each phospholipid is the structure is the lamellar. Lamellar is also called as the leaflet. Hydrophobic fatty acid chains is the minimize their contact with the water by aligned themselves is the together the center of the bilayer forming thick hydrophobic core. Next one, ionic and the H bond stabilize interaction the phospholipid polar the groups with the another with the water. Next one, the phospholipid bilayer is especially does not allow the many water solids, especially the lipid bilayer. They cannot transmit to the some of the lipid particles. Phospholipid layer is that does not allow the many water soluble solutes through the salts, sugars, some small hydrophobic molecule including the water. What are the hydrophobic and the van der Waal interaction between the fatty acid chains? Next one is the all phospholipids bilayer form the sealed closed components inside of the separated from the outside. Maintain the integrity of the bilayer structure. Integrity of the bilayer structure. These are the much more integrity than the outside part of the phospholipid bilayer. The hydrocarbon care the bilayer exposed to the aqueous solution is unstable. Next one is the bimembranes contain the three classes. One is the mainly three classes of the amphipathic lipids. One is the sterol, which is also called as the cholesterol, and the sphingolipids. Next one is the phosphoglycerides. Phosphoglycerides is the derivatives of the glycerol 3 phosphate. They are the consist of the hydrophobic tail composed of the two of the fatty acid based the chain estified of the two hydroxyl group in glycerol phosphate. The polar head group is the attached to the phosphate group. Phosphohydrolates is the head groups of the consist of the choline and the very change of the alcohol and estified the some of the very change the phosphate. Next one is the phospholipase act as the phosphoglycerides. The produce the lysophospholipids. Okay, what are the importance of the lipids, the phospholipids? The important is the signaling of molecules. Signaling of molecules and the release from the cell and the recognized by the specific receptor and their presence of the affect physical properties of MEM. And next one is the sphingolipids. And the sphingolipids are the spinolipids, and the sphingolipid is the derived from the sphingosine. That the derived from the sphingosine. That they have the phosphate-based polar and the head phosphocholine attached to the terminal hydroxyl group of the sphingosine. The terminal, the outer structure, the terminal end of the portion, they can attach to the sphingosine. Okay, next one is the spinoglycomin is the phospholipids and the similar to the phosphoglycan. Some of the spignolides are the amphipathic glycolipids, whose the polar head groups are the sugars that are the linked via phosphate group. Next one is the glycerolipids. These are the three types of the lipids. Already sterols and cholesterol, and the spignolipids, and the phospholipids. The next one is the the next one is the glycosides, and the simplest spinolipids are the contains the single glucose unit attached to the spinoglycin, and the next one is the gangliosides, one or two branched the sugar chains containing the silicic acid groups are the attached to the spinoxin. And next one is the third one is the cholesterol. These are the sphingolipids. All are the sphingolipids. Next one, cholesterol. Cholesterol continues to the third of the membrane lipids, the sterols. Are the four ring based of the hydrocarbons. Cholesterol is the hydroxyl substance on one ring. Major animal sterol. And next one is the some are the amphipathic because hydroxyl group mainly interact with especially the sterol, the glycogen, the cholesterol also is the glycogen mainly reserve materials, especially uh, glucose they can convert the in glycogen are the heavy fat, especially they can convert the cholesterol, glycogen. Next one is the cholesterol abundant is the phospholipids, the mammalian, especially these are the storage of the mammalian present in the prokaryotes some of the plant. What are the prokaryotes? Especially bacteria. Bacteria is the prokaryotes. These are the reserve food material is the glycogen. Okay, these are the cholesterol. Okay, glycogen. And next one, cholesterol is the hydrophobic because from the bilayer structure. These are the bilayer structure. Sterols are the provide the structural membrane, preventing to close the packing of the phospholipids. Acyl chains is the maintain. Acyl chains also maintain the significant measure of the membrane fluidity. The cholesterol is the precursor of the several important bioactive membranes, especially mainly glycogen is the one type of the cholesterol, including the bile acids is made of the in liver mainly. The help of the emulsification dietary fats are the digestion of the absorption of the intensity. These are the membrane lipids. Okay, these are the membrane proteins. Some are the membrane proteins. Three types of the membrane protein. Mainly these are the lipids, phospholipids by layer, liposomes. These are the mainly liposomes, and next one is the some are the spherical micelles. Next one, different types of the lipids also. Lipids, especially membrane, the contain of the three amphiphilic lipids, the sterols, cholesterol, they are also called as sterols, sphingolipids, and the phosphoglycolipids are the membrane proteins. Okay.